understand some conventions for determining anatomical direction. And when we talk about a head-to-tail direction, we're talking about rostral or anterior or caudal or posterior. Caudal is more toward the tail, sort of toward the posterior, and rostral really means more toward the beak or more anterior. And then the belly to back direction would be the dorsal side toward the back and ventral toward the belly. So superior or dorsal are often used synonymously, and ventral or inferior are often used synonymously. Sometimes instead of ventral, the word basal is used. And then when we talk about the relation to the midline, anything that's near the midline of the body is called medial, and anything away from the midline is called lateral. And then when we're talking about the relation of a part of a structure close to or away from its point of origin, a structure could be located proximal to another structure that's near its origin, or it could be located far away from its origin, in which case we would say it's a distal structure or further away. So many times we talk about directions in these using these conventional terms. When we look at brain scans and various images of the brain, there are usually three planes that we look at the brain from. What's called a coronal or transverse image, which is parallel to a vertical plane through both ears. So sort of this slice right here, and if we were to sort of make that slice and then turn the brain around to see what it looked like, we would see something like this. This is called a coronal or a transverse section. A sagittal or longitudinal view is at a 90-degree angle to the coronal plane in a vertical direction. So this is the sagittal plane, where we're sort of taking slices along the sides of the brain from right to left or left to right. So that would be what a sagittal image would look like. And a median or midline sagittal section would actually divide the brain into its two hemispheres. So that's another way to kind of keep in a midline sagittal section would produce a right hemisphere and a left hemisphere. And then a horizontal or axial section is at right angles to the two other types of sections. And so this is the horizontal or axial plane, where we're sort of going from bottom to top or top to bottom. And we can also do the same type of procedure with the spinal cord as well. This is a horizontal or axial section of the spinal cord, and you can see this sort of butterfly-shaped contents within the spinal cord. A couple of other things with nomenclature. Ganglion and nucleus refer to exactly the same thing, except that the ganglion is the first collection of axons or nerve cells outside the spinal cord, and a nucleus is the collection of axons or nerves inside the central nervous system. So we have two different terms for the same thing depending on whether we're inside the central nervous system or outside the central nervous system. And similarly, a nerve, when we talk about nerves, we're talking about a bundle of axons outside the central nervous system, i.e. the peripheral nervous system. And a tract is the same thing, a bundle of axons inside the central nervous system. So when we refer to ganglia or a ganglion, we're talking about a collection of nerve cells outside the spinal cord, outside the central nervous system. The nucleus is a collection of axons or nerve cells, neurons. It should be really neurons rather than nerve cells inside the central nervous system. I mentioned at the beginning of class that we have two nervous systems. And we've talked about the central nervous system being composed of the brain and the spinal cord, and the peripheral nervous system composed of the cranial and spinal nerves, cranial being toward the cranium up here, and spinal nerves coming down the spine, and peripheral ganglia, again, collections of nerves, of cells, cell bodies outside the spinal cord. Peripheral nervous system nerves project to target organs and to muscles, and we call those efferent nerves. And then these nerves also carry sensory information to the brain, afferent nerves, or sensory neurons. And again, remember, sensory, afferent, motor, efferent. Now, the peripheral nervous system can then be further divided into the somatic nervous system and the autonomic nervous system. The somatic nervous system includes the spinal nerves and the cranial nerves. And both the spinal and the cranial nerves have both afferent nerves and efferent nerves. Afferent nerves from the sense organs and efferent nerves to muscles. The autonomic nervous system can be broken down further into two sections, the sympathetic branch and the parasympathetic branch. So the sympathetic branch is the thoracolumbar region. So it's on the spinal cord between the thoracic vertebrae and the lumbar vertebrae. The vertebrae go cranial, thoracic, lumbar, sacral from top to bottom. So the two branches of the spinal cord in the middle, the thoracolumbar section, give rise to the sympathetic branch of the autonomic nervous system. And the parasympathetic branch, or the craniosacral system, involves four of the cranial nerves, nerves 3, 4, 9, and 10. 3, 7, 9, and 10. And the spinal nerves that are in the sacral region, and parasympathetic ganglia that are adjacent to target organs very far from the spinal cord. So again, keep in mind, if you had to describe the organization of the peripheral nervous system, this would be kind of the hierarchical outline that you would follow. Now, the cranial nerves, there are 12 cranial nerves, and they have a variety of functions. Cranial nerve 1 is the olfactory nerve. It's involved in smell, and it's kind of almost right above the eyes on the undersurface of the brain. They're little bulbs, sort of. And then the optic nerve is involved in vision, carries vision from the eye back into the brain. The oculomotor nerve, nerve 3, is associated with eyelid and eyeball movement, and it innervates the superior oblique muscles in the eye. I'm sorry, innervates the superior oblique. Right? Eyeball and eyeball movement, where am I? Yeah. Trochlear innervates the superior oblique muscle, and the trigeminal nerve turns the eye downward and laterally. Trigeminal nerve also is involved in chewing. The abducens nerve, nerve number 6, is associated with face and mouth sensation, both touch and pain. The facial nerve has a lot of functions. It turns the eye laterally, controls most facial expression, and is involved in secretion of tears and saliva, and participates in taste sensation as well. I think we talked about Bell's palsy, and that's an infection of the 7th cranial nerve that causes a paralysis of one side of the face, kind of a drooping of the eyelid. The vestibular cochlear nerve, or simply the auditory nerve, is involved in hearing and equilibrium. Glossopharyngeal nerve involved in taste and salivation, and it's also involved in sensing carotid blood pressure and providing feedback to maintain blood pressure. The vagus nerve Vagus means wandering, and it actually travels very far from the spinal cord, being a parasympathetic nerve. It senses the blood pressure in the aorta, is involved in slowing or kind of regulating the heart rate, but specifically slowing it if it's stimulated. It stimulates the digestive organs and also is involved with taste. Spinal accessory nerve controls the trapezius and the sternocleidomastoid muscles, so muscles kind of in the upper shoulder area. Spinal accessory also controls swallowing movements. So trauma to that would affect somebody's ability to swallow or to eat. And the hypoglossal, or 12th cranial nerve, controls tongue movements. Yeah?
If someone has like tics or something, would that mean that one of those areas is affected? Like if they have like scar, like a facial tic or something, is that like one of those nerves?
Normally not. That has an involuntary movement like that. Usually there's actually on the next slide there's a little link that you can go to see testing of the function of each of these different cranial nerves, like a neurologist might do. But in that case, there's actually a different process that's associated with that. It's not specifically the dysfunction of cranial nerves. Yeah. There's a variety of ways to remember the cranial nerves, the names of them. You know, there are 12 of them, and so various mnemonics have been developed, some more memorable than others, but the one I usually use is on old Olympus, towering top, a Finn and German viewed some hops. So each of the first letters correspond to the first letters of each of the 12 cranial nerves. But most importantly, 3, 7, 9, and 10 are your parasympathetic cranial nerves. And you'll notice that some of the cranial nerves have exclusively sensory functions, some have exclusively motor functions, and some have mixed functions, both sensory and motor. So this diagram, the red and blue will designate whether one of the cranial nerves is either sensory only, motor only, and the others, I think in black, are mixed. And there's, again, 12 pairs of cranial nerves, one on either side. So spinal nerves include 31 pairs of spinal nerves. There's 8 cervical spinal nerves, 12 thoracic, 5 lumbar, 5 sacral, and 1 coccygeal nerve. And if somebody has a slipped disc or a disc compression in their back, for example, there's a little insulation between the bony vertebrae in the back. And sometimes as we get older, that cartilage or that substance that separates and cushions the vertebrae from each other starts to dry up. It desiccates. And so sometimes with severe back strains, the disc can actually push out and cause severe compression of the nerves, which will result in various phenomena like pain or difficulty moving or difficulty with sensation, unusual sensations, or numbness or tingling, things of that nature. But 12 cranial nerves, 12 on each side, 31 pairs of spinal nerves. And this is an overview of the autonomic nervous system, and one of the major things that kind of jumps out is in this thoracolumbar, or the middle 2 sections of the spinal cord, we have the sympathetic nervous system. And the sympathetic ganglia, the first collection of axons outside the spinal cord, is organized in a neat chain right close to the spinal cord. And then that's the preganglionic fiber, the fiber between the spinal cord and the first ganglion. The postganglionic fiber is what comes afterwards. And so the postganglionic fiber for the sympathetic nervous system is very long and goes to very diverse distant sites. By way of contrast, if we look at for example, the parasympathetic, the cranial nerves up here, and the sacral nerves, craniosacral system of the parasympathetic nervous system, we can see that the preganglionic fiber isn't organized in a nice little chain. It's organized where there's a long, distant preganglionic fiber and a very short postganglionic fiber that synapses right on the organ that's the target of stimulation. So that's kind of a big difference between the organization of the sympathetic and the parasympathetic nervous systems. We have a neat sympathetic chain for the sympathetic nervous system versus a very diffuse distribution of the ganglia for the parasympathetic nervous system. And we see some other differences as well. We know that the sympathetic nervous system originates in the thoracolumbar region of the spinal cord, whereas the nerves, parasympathetic nerves, originate in the cranial and the sacral region. This is just another diagram that kind of compares the sympathetic and the parasympathetic nervous systems. We often kind of refer to the sympathetic nervous system as the fight or flight system. It's the one that really revs us up and gets us ready to either fight in an aggressive situation or to run and to get out of there, and gives us energy to the muscles, shuts down things like digestion and things of that nature. In the parasympathetic division, we call that the rest and digest system. So when the threat is gone, we don't have to flee or fight, then we can rest and digest our food. So there's a lot of activity with slowing the heart rate to rest. That's the parasympathetic nervous system taking over through the vagus nerve. And enhancing digestion, stimulating more blood flow away from the muscles into the digestive organs. So as I mentioned, the differences between sympathetic and parasympathetic nervous systems are important to know. So the ganglia types, we have early ganglia in the sympathetic nervous system. And there's very short fibers that leave the spinal cord to form the ganglion. And the parasympathetic nervous system, we have distant ganglia. The fibers are very long, and they leave the spinal cord and travel quite a distance right to the target organ before forming the ganglion close to the target organ. The distribution of ganglia is in a neat sympathetic chain for the sympathetic nervous system, but there's a diffuse distribution of the ganglia in the parasympathetic nervous system. The sympathetic nervous system has very diffuse targets. The long fibers leave the neat ganglia chain and branch all over to many targets, whereas the parasympathetic targets are actually local to the target organ themselves. And two more differences: the neurotransmitter involved. For the parasympathetic nervous system, both the preganglionic and postganglionic fibers are cholinergic. They use acetylcholine. But in the sympathetic nervous system, the preganglionic fibers are also cholinergic, but the postganglionic fibers are noradrenergic for the most part. The one exception is the sweat glands, which are cholinergic. Also, in the sympathetic nervous system, the adrenal medulla is innervated directly by preganglionic fibers. There is no postganglionic fiber for the adrenal medulla. And then the location, the cell bodies of sympathetic motor neurons are located in the thoracic and lumbar regions of the spinal cord, and the cell bodies that give rise to the parasympathetic axons are located in the nuclei of some of the cranial nerves, like the vagus nerve, and also in the sacral region of the spinal cord, the craniosacral system. Important point, not all cranial nerves are involved in the parasympathetic nervous system. Only 3 of them are the parasympathetic nervous system. Actually, only 4 of them are involving the parasympathetic nervous system. So again, nerves 3, 7, 9, and 10. So moving from the peripheral nervous system into the central nervous system, we have one of the most important, or probably the most important, what brings us here today is the brain. And the brain weighs about 400 grams when we're born, and it doubles, more than doubles in size in less than a year, up to 850 grams. And it nearly triples in size from birth weight at age 3. Then the size of the brain plateaus at about 1,250 to 1,450 grams by maturity. And unfortunately, once we pass age 30, the brain starts to shrink. And by about age 75, the brain shrinks by roughly about 100 grams. Some of my studies involve centenarians, as I mentioned, or 100-year-olds. And when we weigh their brains after living 100 years, they're around about 900 grams or so on average. So again, losing quite a bit from that 1,250 to 1,450. The brain is made up of 3 different types of matter, gray matter, white matter, and glial cells. The white matter are those myelinated fiber tracts within the brain that we talked about earlier. And white matter doesn't involve it doesn't consume a lot of energy. White matter consumes only about 6% of the cerebral oxygen supply, but gray matter is where really most of the oxygen consumption takes place. Gray matter consumes about 94% of the cerebral oxygen supply, and there's about 100 billion neurons in the gray matter. We talked about the glial cells earlier. As I mentioned, they provide structural and metabolic support, immunologic support, and there are about 10 to 50 times as many glial cells as there are neurons. As you know, the brain has 2 distinct hemispheres, right and left. And the hemispheres are held together by a band of white matter fibers called the corpus callosum. The brain gets about it represents only about 1/50th of our total weight, but it consumes about 20% of our total blood flow from the heart. And it receives that 20% constantly. So whenever there's shifts in the flow of blood to various organs or muscles or so forth, the brain always gets the constant blood flow. The primary food of the brain is glucose. It can't store glucose, unfortunately, but again, astrocytes convert the glucose to glycogen, and the astrocytes then can store the glycogen that can be later converted into energy. Just a 1-second interruption of blood flow to the brain uses up most of the dissolved oxygen in the blood. And a 6-second interruption of blood flow to the brain will produce unconsciousness, and permanent brain damage will actually begin within a few minutes after blood flow ceases to the brain.
What causes the interruption?
If there is an interruption from, for example, a hemorrhage, or well, it would be probably a bigger hemorrhage of losing a lot more blood, or if somebody is suffocated, for example, or things of that nature. There are 5 parts of the brain that you should be familiar with. And we have the telencephalon, which involve the cerebral hemispheres and subcortical structures that are called the basal ganglia. Those are important in movement, for example, and in what's called procedural memory, or learning how to do a task, like that mirror tracing task that I mentioned with HM. The diencephalon consists of the thalamus and the hypothalamus. The mesencephalon is also referred to as the midbrain. And this includes the superior and inferior colliculi. The superior colliculi are involved in very quick visual movements. If there's a flash of light, my gaze suddenly is directed over there. That's the superior colliculi an operation. And if there's a loud sound out in the hall or something and I orient toward that, that's the inferior colliculi working. The red nucleus is involved in motor functioning, as is the cerebral peduncle. The cerebral peduncle includes the crus cerebri, the substantia nigra, and the tegmentum. The medencephalon includes the pons and the cerebellum, and the myelencephalon includes the medulla oblongata. The last three, the mesencephalon, medencephalon, and myelencephalon, collectively are referred to as the brain stem. So if there's damage to somebody's brain stem, then it involves some kind of a problem in one of those three regions of the brain. That's a very simplified breakdown of the 5 different parts of the brain. We have our telencephalon, which includes the cerebrum, the diencephalon, which is the thalamus and the hypothalamus. Basal ganglia also included in the telencephalon. The midbrain is represented here. The pons and the cerebellum, so the medencephalon and the myelencephalon, or the medulla down here. And then the medulla then leads into the spinal cord, which is distinct from the brain, what part of the central nervous system. This again is sort of a sagittal view of the brain, where we have the nice image of the corpus callosum and one hemisphere of the telencephalon. Shows you the cerebellum and the pons very nicely, midbrain up here, and medulla oblongata down here. And the foramen magnum, or large opening, is where the medulla leads into the spinal cord.
Is that the pineal gland?
This thing?
Yeah.
No, that is this is the thalamus, and that is a little band of fibers that connects the two sides of the thalamus. So the telencephalon includes the center for the highest brain functions or highest cognitive functions. It's most developed in humans compared to other animals, and it consists of the 2 cerebral hemispheres. We look at the outside of the brain and we refer to gyri, or these little bulges here, and sulci, or these little grooves. And a deep sulcus, singular, is called a fissure. And so we have the fissure of Rolando here. This is called the central fissure also, and it's very important for separating the frontal lobe from the parietal cortex. We have another horizontal fissure called the fissure of Rolando, fissure of Sylvius, or Sylvian fissure. That separates the temporal cortex from the frontal and parietal cortex. So those are really 2 important fissures that you should be familiar with, the fissure of Rolando and the fissure of Sylvius, and that serves to demarcate most of the lobes of the brain. The only other one is the calcarine fissure, that's a little bit harder to see in the back of the brain, but that separates the occipital cortex from the parietal and temporal cortex. Now also, each of these gyri and sulci have names, and I find the names pretty convenient for the most part, because again, following our convention for naming anatomical direction, for example, here in the temporal lobe, this gyrus would be called the superior temporal gyrus. It's the gyrus in the temporal lobe that's on the top. And down here we have the inferior temporal gyrus. It's the gyrus in the temporal lobe that's on the bottom. And unbelievably, this one is called the middle temporal gyrus, because it's the gyrus in the middle. So they really helped us out with naming conventions for once. Seems to be relatively straightforward. We also have superior frontal cortex, middle frontal cortex, and inferior frontal cortex. There's a couple of other little gyri that have little less conventional names, but for the most part, they kind of follow that pattern. So in the telencephalon, we have the gray layer, which is the outside cortex, made of cell bodies, glial cells, blood vessels, and then the myelinated axons that comprise the white matter. There's no cell bodies, but a substantial amount of neuroglia, but fewer capillaries than gray matter. The glial cells in the inner white matter are predominantly oligodendroglia because they're forming the myelin sheath. The inner white matter can be divided up into three different types of fibers. Commissural fibers, which pass from one hemisphere to the other, they help to connect the hemispheres together. And the associative fibers pass from one lobe to another or from one gyrus to another within the same hemisphere. So they connect specific regions of the brain within the same hemisphere together. And then projection fibers are afferent and efferent tracts, sensory and motor tracts, that connect the brain to the spinal cord. And so a lot of the projection fibers are situated in a region of the brain called the internal capsule, which has an anterior part or anterior limb, a posterior limb, and a middle genu. Genu is knee. It's a bend like a knee might be. And we can see the internal capsule here. This is the anterior limb of the internal capsule. It curves around. Here's the posterior limb of the internal capsule. And here is the bend or the genu or the knee. Moving to the diencephalon, we have the thalamus, which is the main sensory relay center, main sensory way station, if you will. The hypothalamus, which is involved in temperature control, emotional states, and control over the autonomic nervous system. So feeding, fleeing, fighting, and mating are the predominant functions of the hypothalamus. And then we have some other secondary structures that are also considered part of the diencephalon, which are the medial and lateral geniculate bodies, part of the thalamus. Medial geniculate body being more an auditory processing center, a lateral geniculate nucleus associated with visual sensory processing, and then the pineal body and the habenulae. Together they're called the epithalamus. We talked about the pineal body as being the only structure in the brain that doesn't have distinct hemispheres, and the habenulae are attached to the pineal body, and they're involved in the reward system. They have both serotonergic and dopaminergic neurons. The mesencephalon is the smallest of the 5 divisions of the brain, and this is the area above the aqueduct of Sylvius. This is called the tectum or roof. The tectum is made up of what's called the corpora quadrigemina, or the superior and inferior colliculi that I mentioned earlier. You have one on either side, and corpora quadrigemina means 4 bodies. So you have 2 superior colliculi, 2 inferior colliculi, which make up the corpora quadrigemina. The tegmentum, or body, contains fiber tracts, the red nucleus, oculomotor nerve and trochlear cranial nerves and their nuclei. The substantia nigra is involved in movement. It's a very black pigmented substance in the brain. It becomes very pale in people with Parkinson's disease. So normally it has a very black color when you look at it directly, but due to the degeneration of dopaminergic neurons in Parkinson's disease, that becomes more pale. And then the crus cerebri is a pair of huge fiber bundles that continue the descending projection fibers from the internal capsule that I mentioned earlier. Collectively, the substantia nigra and crus cerebri constitute what are known as the cerebral peduncles. So there's a lot of stuff going on in the midbrain, a lot of different portions for a very small region of the brainstem. The metencephalon includes the cerebellum and the pons. Cerebellum constitutes about 10% of the brain's mass, but amazingly, 50% of the brain's neurons in this relatively small structure in the back of our head, right behind our cerebrum. is divided up into the archicerebellum, or older cerebellum, the paleocerebellum, and neocerebellum, and then several different nuclei that are involved in movement. You don't have to know the specific segments of the cerebellum, but if I asked you to tell me what constitutes the medencephalon, you should be able to tell me the cerebellum and the pons. The pons is separated from the cerebellum by the fourth ventricle, and there are a lot of ascending or sensory tracts and descending or motor fiber tracts that pass through the pons, and it contains the nuclei of several cranial nerves, specifically 5, 6, and 7. Trigeminal, abducens, and facial nerves. The medulla oblongata becomes contiguous with the spinal cord at that large opening that I showed you earlier called the foramen magnum, and it contains the ascending and descending fiber tracts as well for sensory and motor functioning respectively. And here in the medulla we find the nuclei for cranial nerves 8 through 12. And the medulla contains respiratory and cardiac centers. Lesions or damage to these regions can cause coma and death. When we look at specifically the cortex, we have 6 principal gyri within the frontal lobe that are associated with various functions. The precentral gyrus is called motor cortex. We call it precentral because it's the first gyrus right in front of the central fissure that we talked about earlier. That's responsible for your primary motor function. It's anterior and parallel to the central sulcus, or fissure of Rolando. And neurons here give rise to corticospinal and corticobulbar tracts, more involved with movement. Next, we have the superior frontal gyrus that's concerned with body and eye movements. And so that's just a little bit anterior. The middle frontal gyrus, superior middle frontal, concerned with also body and eye movements. And inferior frontal, which is down here, that includes Broca's area that we talked about last week. The orbital gyrus is right above the eyes, and that's important in the expression of emotion. And then there's an area called the gyrus rectus right here, kind of a straight gyrus, and that's also involved in reward and reinforcement. In the parietal cortex, we have 2 major sulci, the postcentral sulcus, which extends behind and parallel to the lateral fissure or central fissure, and it consists of a superior and inferior portion, and then what's called the interparietal sulcus, the horizontal groove that sometimes connects with the postcentral sulcus. It's a little harder to see the interparietal sulcus. The postcentral gyrus, or sensory cortex, it's that strip of, or that gyrus that lies immediately behind the central fissure, or fissure of Rolando. And that's associated with the appreciation of touch, or somesthesis, sense of position, of extremities, or kinesthesis, vibratory sense, and other fine tactile discriminatory processes. We can divide the parietal lobe up into 2 sections, the superior parietal lobule and the inferior parietal lobule. And it's in the inferior parietal lobule that we find the supramarginal gyrus and angular gyrus, which are concerned with reception and organization of language functioning. And you can see them in this diagram here. It's a little bit harder to read from your chairs, but you'll probably be able to see it on the course caption. The occipital lobe is pretty straightforward. We have